Hello and welcome to my channel. In today's video, we are going to discuss about biotechnology. So biotechnology is basically a branch of biology which deals with biology and technology. That is how biology deals with utilization of living beings or living processes for production of products which are important for or useful for humans. It is the use of an organism or a component of an organism or other biological system to make a product or process. Since it uses DNA, most important fact in biotechnology is recombinant DNA biotechnology. So DNA technology is the sequencing, analysis, and cutting and pasting of DNA. Beer brewing is one of the examples where fungi are introduced into a solution of malted barley sugar. It undergoes a process called fermentation, and the byproduct of the fermentation is the alcohol that's found in the beer. Penicillin is one more example. The antibiotic penicillin is generated by molds, a mold being used to make a product of human use. In DNA technology, it depends on the ability to analyze, manipulate, and cut and paste pieces of DNA. It can be used to make copies of a DNA sequence by a technique called PCR, Polymerase Chain Reaction. This I have already explained in my previous video in detail, and I will put that in my description box. Link of that video. Next is DNA cloning. Many copies of a DNA fragment of interest, such as gene. So gene is basically functional unit of DNA. DNA is blueprint of life, which holds all the information of an organism. DNA cloning involves inserting a target gene into a circular DNA molecule called a plasmid. Plasmid is a vector. It's like a carrier molecule which helps in transport of the target gene from the source to the host. Talking a little bit about the technique polymerase chain reaction PCR, it is a reaction which produces many copies of a target DNA sequence starting from a piece of template DNA. And this technique can be used to make many copies of DNA. So this is how DNA duplicates. Duplication of DNA is done. Next is gel electrophoresis. In gel electrophoresis, DNA fragments are separated based on their size and fragments. They are stained with a dye. This technique is used to visualize DNA fragments. This is a gel. This is a ladder which has different molecular weights. Different molecular weight sample and DNA fragments on it. DNA sequencing. It involves determining the sequence of nucleotide bases, ATGC, in a DNA molecule. In gene cloning, the foreign DNA, whatever particular portion of this DNA we want, we can just cut that particular portion, take a plasmid, cut a particular portion of the plasmid, insert the gene which you need to transport into this plasmid with the help of the restriction enzymes. This cutting is done. The restriction enzymes are like molecular scissors. For example, EcoR1, GATC, CTAG, and plasmid DNA carrying foreign body, and it's called ampicillin resistant gene, this marker which helps in transport properly. So this foreign DNA fragment with the help of plasmid is transferred to the bacterial host. So in gene cloning, basically we have three DNA: foreign DNA, the DNA of interest, the particular fragment of this DNA. So first of all, what is done? With the help of restriction enzymes, this foreign DNA sequence will be cut and this fragment will be inserted here in the plasmid DNA. And then this plasmid DNA will be transported inside the host along with the gene of interest. And this is how transport, gene cloning is done. The foreign DNA and plasmid are cut with the same restriction enzyme which recognizes a particular sequence of DNA called a restriction site. The restriction enzyme creates sticky ends that allow the foreign DNA and cloning vector to anneal, and an enzyme called ligase glues the annealed fragments together. It is the process of isolating and making copies of a gene, and gene cloning might be used to isolate and make copies of a normal gene for gene therapy. This is done by putting the gene in a bacterial plasmid, and plasmid DNA, which is circular, that is not part of a chromosome and can replicate independently. These are the steps: isolation, ligation, transformation, and selection. That's all for the video. Thanks for watching.